Hi everyone, good afternoon. Thank you all for joining us. My heart is with you all. And my name is Tal Gilboa. I'm co-founder of Everest Biolabs. And today I'll present you our products, and I thought to make it more interesting and also show you a little bit about my EV research, how I got into the EV field, and the tools we have to support your EV research as well.
Tal, we see when you share the screen, we see us as well. So you
Really?
can minimize. Yeah, maybe you can minimize it.
Like that?
Yeah, great. Yeah, great. Thanks.
Okay. Now you, okay,
Yeah.
awesome. Sure.
Oh, yeah. Okay. Yeah.
Looks good?
Yep.
Okay, cool. So just one slide about our big vision. So we aim to develop basically an ecosystem for all scientists and companies working on EVs by giving tools and developing technologies to solve the entire workflow for EV research. So how to isolate exosomes in a reproducible manner. We work a lot about automation, how to do this isolation automatically with high throughput, specifically for clinical studies. And also after you isolate your EVs, how to analyze them and look at different EV biomarkers. So a really big goal is to advance diagnostics, therapeutics, and to support clinical trials using EVs. And just a little bit about how I got into the EV field. So my postdoc was focused on Parkinson's disease. My goal was to develop new diagnostic tools for Parkinson's or PD. So PD is currently diagnosed clinically. And one of the reasons we cannot diagnose this disease better is that it's very hard to see what's happening in the brain of living patients. We can't just easily biopsy the brain. But if we could see what's happening in the brain, we would see that neurons are slowly dying. And so in Parkinson's, we have this protein called alpha-synuclein, which misfolds and aggregates, resulting in neuron death. So we wanted to develop new tools to find this early aggregation or pathology of Parkinson's. And this is how we came to EVs, because I'm sure all of you know. And by the way, just stop me for questions if you have any time. So EVs are released from all of our cells to the circulation, including the brain cells. And as they release from the cells, on their surface, they have these markers which are specific to the cell of origin, and they are sampling the cells. And so we can really use them to see non-invasively what's happening in specific tissues. And so my goal was to use brain-derived EVs and other markers for Parkinson's. So the workflow we were developing was to take patient samples, which can be blood samples, CSF samples, and then isolate specifically the EVs coming from the brain, and then burst them open and look for specific markers which could help us diagnose Parkinson's and track disease progression. So we began to develop technologies for each of these steps in this workflow. And specifically, we realized how all this workflow starts with the EV isolation. And so when you isolate EVs, you have lots of things you need to consider. Usually for diagnostics, we want to start with, let's say, less than 1 ml of biofluid, which can be ideally plasma or serum, non-invasive, or urine, and then also CSF. And for some application, you're using cell culture. You want to isolate these EVs from all the other things in your biofluid. Specifically, plasma has a lot of proteins, lipoproteins. So you want to get these EVs out. And your isolation depends on your downstream analysis. So for some application, you want very pure EVs. For example, if you're doing unbiased mass spec analysis. For some application, you just maximize their EV yield. For example, I was looking for these very rare EVs coming from the brain. So I wanted to maximize my EV yield. And for clinical studies, when you want to work with, let's say, hundreds or thousands of samples, you really think about the throughput, reproducibility, and automation, because isolating the EVs one sample at a time for 1,000 samples can be very laborious and also not so reproducible. And so we realized that if we want to optimize how we isolate the EVs, we have to somehow quantify them so we can compare different isolation methods. And so we developed these very sensitive immunoassays based on the Simoa technology, which was developed in the lab I was doing my postdoc at, where we quantified tetraspanins on the EVs. For those of you who don't know, tetraspanins are proteins which pass the membrane 4 times, so they're very membrane specific, and they're abundant in all the cells in our body. So we developed assays to quantify what we call EV proteins, as well as contamination, so albumin and proteins on lipoproteins. And using these assays, we can then start comparing different methods for isolation and look at the ratio between our EVs to our contamination. And while we did that, we began to just explore lots of different isolation methods. And you can see here on the right how the data would look like. You will have your tetraspanin results, which represent the EVs, and the contamination results. And you can look at the ratio between the EVs to the contamination. And so here at Everest, we have very similar assays to look at EVs. We call it the Atlas ELISAs. Those are very simple one-step ELISA. You basically take your sample, you add it to our plate, and it can be, the plate comes in strips, so you can run 8 samples at a time or up to 96. You add your sample, and your sample can be purified EVs or biofluids. So this assay works with plasma, CSF, serum, and all biofluids, and cell culture, of course. And then you add our reagents, which are specifically labeling and capturing tetraspanins on intact EVs. Then you have a 1 incubation step, 1 wash step, and you're just adding the substrate. And this is an absorbance-based assay. So very simple one-step ELISA where you can quantify the EV, and you can use that to QC your isolation and to compare different plasma or sample isolation to make sure you have a reproducible workflow. So using these ELISAs, we realized how one of the best methods, in our opinion, for isolating EVs in biofluids, 1 second, someone is trying to get in, is to use size-based separation or SEC. I'm sure most of you know about this method. It's been here for a long time. So just quickly, the way it works, we have this column which we pack with resin. And this resin is just like big beads which have pores inside. And then you can add your sample on top of the resin. It will start flowing through the column. And all the small particles, which are smaller than the pore diameter on the resin, will be trapped in the resin and interact with it. So they will go slower through the column. And all the large particles, like the EVs, will just go directly down because they are too big to interact with the resin. And so we can start collecting fractions where the first fractions will contain all the large particles, specifically the EVs, and the later fraction will contain the smaller particles like the proteins. And so you can see here how would the elution profile look. So if we're measuring the EVs, they will come in the first 3 fractions, and then the secreted proteins will come later. And the reason we liked this method from all the other methods is first, we could really optimize the yield and the purity in this isolation, and we got to a point where we're getting the majority of the EVs from the sample. It's a very gentle method, so you have methods like precipitation, where you have to resuspend your pellet, or ultracentrifugation, which expose the sample to high forces. This is a very gentle method, so it preserves the EV integrity as they go through the resin. And as engineers, we realized how this method is really scalable. So it's really amenable to automation. So fits our goal of looking at a lot of samples. And so one of the things you can control here is your resin itself, and specifically what is the diameter of the pore in the resin. And you have this trade-off. So the smaller the pore of the resin is, you will get more of the EVs, because you know EVs can be as small as 30 nanometer and as large as hundreds of nanometer. So if you have too big of pore size in your resin, you're trapping all the small EVs. So you can see here, if you have 70 nanometer, which is the purple line, you'll get much less EVs. You can see the EVs in the solid lines here. Then if you have a smaller pores, but then the trade-off is, which you can see in the dotted line, that you will start having the small proteins coeluting with your EVs as well. So the trade-off is between the purity and the yield with this pore diameter. And again, you need to think about that in the context of your analysis. So if you want very high yield EV isolation, you would take the smallest pore size and compromise a little bit of contamination. And so here in Everest, we have two different columns, which we call the apex columns. One has pore size of 35 and one 20 nanometer. So 20 will give you the majority of EVs from your sample, which you can see here in the orange line. With the small contamination of free proteins. And then as we increase the pore to 35, you'll get a little bit less EVs, around 15% less EVs. It's all those very, very small EVs which will interact with the resin, but they will be very, very clean in terms of free proteins. And one of the important things for us is to optimize these columns for clinical work, where it's really important how all the columns will behave the same. So we work a lot on reproducibility of the columns. You can use the columns multiple times. We tested up to 5 times. And these columns work with a typical clinical sample volume, which is between 0.5 to 1 ml. Any of you who's working with smaller sample volume, for example, I know for CSF, usually the aliquots are smaller. We are releasing smaller version of these columns, which will fit the small input volume, and we have an early access program, so feel free to speak with me after the talk if it's something you're interested in. And we've characterized the EVs using these columns from lots of biofluids. This is just an example of NTA, TEM, single EV imaging using cell culture and plasma, but they work with all biofluids. Again, the advantage is that it's a very gentle isolation, so works with any sample. And again, one of the things we really care about is reproducibility. Because if your columns are packed slightly different, let's say the compression percentage is different or the amount of resin is different, the EV elution profile can shift. So you can see here how it looks. If you have a slow column, the EVs will come earlier because you have less of void volume. So they will just shift to the left. And if the column is too fast, the EVs will come later. Because you have more of void volume in the column. And then what happens is that you would isolate your EVs, but have different isolation for different samples. And the way to control it is to make sure that the drip speed of the columns is reproducible, which is something we really care about. Again, I was using these columns for my clinical research with Parkinson samples. So we make sure that the columns elute the EVs in a very reproducible manner. So you don't have to be scared about collecting the wrong fraction accidentally. All the columns will behave exactly the same, which you can see here on the right. Now, just example of how I use these high yield isolation columns in my research. So again, I was interested in alpha-synuclein, which is a protein related to Parkinson's disease. And the question we asked was, what is the upper limit of alpha-synuclein we have in EVs? And so to answer this question, we wanted to isolate the EVs with the highest yield possible. And so we chose the columns with the smallest pore size. which are the apex 6B columns. And then we compared the amount of alpha-synuclein in the biofluid to the amount of alpha-synuclein after the EV isolation. So first we saw how if we're just comparing alpha-synuclein to tetraspanins, tetraspanins again represent the EVs. They would elute in the earlier fraction, which you can see here in gray, whereas the albumin, the free protein, will elute in the later fraction because it's a small protein. And you can see how alpha-synuclein, which is here in blue, has this two-peak distribution. So you have some alpha-synuclein, which is associated with the EVs, and alpha-synuclein, which is just free in circulation. So we could really quantify the amount of alpha-synuclein in EVs, and also we looked at phosphorylation, which is relevant for Parkinson's. And again, we could use these high-yield columns without thinking too much about the purity because we used targeted analysis. We had specific assays looking at alpha-synuclein and the alpha-synuclein phosphorylation. But what happens if you want to look at untargeted analysis, for example, mass spec, where it's actually the opposite? You care a lot about the purity. You want to make sure you're looking only on EV proteins. So in this case, you will have an issue because we also have this lipoprotein in the blood, which there are lots of different lipoproteins. There are high-density ones and low-density ones. And some of them really overlap by size with the exosomes, with the EVs. And so if you're using size separation, you would co-isolate your EVs with some of these lipoproteins. And then in your mass spec, the top hits will be lipoprotein-associated proteins. And so to overcome that, we developed these multi-mode columns. Those are columns which have basically multiple functions inside. So on the top, they have the regular SEC I showed you before. So they're filled with this resin, which traps the small protein. And so we have a separation of the EVs from the small protein. And then as the EVs reach the bottom layer, they have another layer of cleaning. And so in this case, we used cleaning by charge and by hydrophobicity. And so the EVs you get at the end are really, really clean EVs. So the trade-off again, you're losing some yield because some EVs are going to be now trapped in the bottom layer, but you're gaining a lot of purity in terms of both free proteins and lipoproteins. And so here in Everest, we developed last year a commercialized version of these columns. We call them the Apex MM columns or multi-mode columns. They work just like I showed you. They have this two-layer configuration. And what you get is the purest EVs. So this really saves some people traditionally would do multi-isolation. They would do SEC first, then density gradient sometimes. So here you have one column which really cleans the EVs for you, both from the high-density lipoprotein and the low-density lipoprotein. They work, again, with sample input of 0.5 to 1. And we are releasing the smaller input volumes in ISEF next month. If it's something, again, you're interested in, please speak with me. And just to show you the advantage, so basically, if you are using NTA on your EVs, it's a little bit problematic because NTA just measures particles, and lipoproteins are so abundant in the plasma that you might just measure lipoprotein in your NTA. But here on the green, you can see this is work we've done with particle metrics, that if you look at the ratio between our Atlas ELISA, which measures specifically EV biomarkers, to the scatter analysis from NTA, in the multi-mode columns, basically most of the particles you're measuring are EVs because we really cleaned all the other particles. So you're getting really pure exosomes. Sorry. Okay. And if you aren't sure what is the best column for your research, we added this column selector feature to our website, where you can see a list of the different analysis you're interested in, and then it will give you a recommendation of what is the best column for you. So you can check it out in our website. So that was just our columns, and traditionally, historically, people would run them manually. So they would have this holder, put the column, wait for it to wash. You have to really wash these columns well because we store them with antimicrobial reagent, which takes like an hour of wash. Then you would add your sample, wait for it to go down the column, and then start collecting fractions. So it's a lot of work for one sample. And if you want to measure 1,000 samples, it's quite a lot. And you also have like variation because of manual pipetting. And so thinking about clinical studies, automation and throughput was something we really cared about. And we wanted to understand how we can scale and automate this isolation. And so the first instrument we developed in my postdoc, we called it the octosome. It's basically a very simple method to isolate EVs from 8 columns. So you can see how it looked like on the bench. We had a pump connected to a small computer, and the pump will just flow PBS through the columns. And then we could move between collecting the EVs to waste. So that was the prototype. And then here at Everest, we made it a real instrument, which we call the Ascent. So the Ascent is for running up to 8 samples, collecting up to 12 fractions. So you can measure many more samples at a day, minimal hands-on time. The only thing you have to do manually is to add your sample. It will wash the columns for you. Because it's almost fully automated, it will be very reproducible. And just about the way it works, so you have here the holder for the columns. This instrument works with our columns as well as the ISON columns. You have 2 different positions for the columns, for the larger and the smaller columns. And you have, let me put the video again. And you have 2 positions for your collection tubes, again, to have access for the larger columns and the smaller ones. We have here the 3 bottles. This is a buffer bottle, water bottle, and bottle for the waste. And here you have these 8 dispensers, which dispense the buffer on the columns. And then on the bottom, we have drop detectors. They're looking at the drops as they fall through the column, so they will know when to move to the next step. And then we have the collection tubes on a stage, just moving below the columns for collecting the fractions. And so this instrument will wash the columns for you, then it will call you to come in and add the sample. Now, because the idea is that to give you free time to do your other work in the lab, you might not be next to the instrument as it's done washing. So we have this column saver option, where even though it finished washing, the instrument will keep having the columns wet. So the columns are not dry and they're just waiting for you to come with your sample. It will beep for you to come, but the columns will be as good as possible, perfectly ready for you when you come with the sample, and then you just press go and it will do everything. It will wait for the sample to go through, add the void volume, and then collect the fractions. So that's the Ascent, and this is for 8 columns. And again, the only manual step here is to add your sample manually. But 8 columns was just the beginning for us. We were looking at larger scale work. And so we thought, how can we really bring SCC to real high throughput? And the way to do that is to move from a single column format to a plate format, because it's much easier to automate things when they're in the plate format.
Yeah.
Awesome. Okay, so before the break, we were talking about how we wanted to improve our throughput and automation. And so instead of a column, we developed these deep well plates, where it's a 24 plate, like you typically see on the top. But if you look from the side, it's very deep. So basically each well represents one SEC column. and it has enough resin to separate the EVs from the other contamination, just like a column. And once we had the plate format, it was very easy to automate. So in this work, we used just a Tican instrument, an OpenTrans instrument with this plate, and showed how we can run 2 in parallel, so 48 samples, and do it in a fully automated manner. So we have the samples on the deck, we have the plates on the deck, we have PBS, and everything else is done on the Tican. The Tican only has these 8 pipettes, so it's a little bit slower, but it has to do all the washes 1 ml at a time, but still a fully automated process with 48 samples. And so here in Everest, we built these columns as well. So these columns, again, they're 24 deep well plates, so you can see how they're as deep as the column. Each well represents a column, and they work just identically. So the idea is you can, as you are developing, you can work on individual columns, you can use the ascent, or run it manually to just make sure your assay is working and you're happy. And when you're ready to do the high scale study, you can just move to the plate and you don't have to worry about re-optimizing your assay, changing anything, because they work just identically. Someone is raising his hand. Eliran, or is it from before? I can't see the chat, so it's from before. Okay, awesome. It's only before, yes. Okay, great. So to use these plates, we built the Summit instrument. This is a fully automated plate-based isolation and concentration instrument. So the way it works, let me start the video. So we have here this liquid handling robot. It has 4 pipettes, 4 dispensers, and on the back, it has a liquid level detection, which you can see now. So basically, you would put your samples here. Your samples can be in tubes, or you can put them in a deep well 96 plate. And then the Summit will it will first start by washing the plate with the dispensers. After washing, it will load your samples with the pipettes, fully automated. Then it will wait for the discard volume, and then it will start collecting the fraction. So you can collect with it up to 3 fractions. Usually people optimize the fraction collection with a single column or multiple columns, and then they know what is the final elution they want, and we just need 1 fraction for the EVs. And then you can see it now. Basically, when you collect the EVs, you can collect them either on regular collection plate, and the Summit will move it to either an output plate or to tubes, or you can already concentrate. So you can load here ultrafiltration plates, and it has a vacuum system on the back, so it will just concentrate your EVs. You will tell the Summit what is your final volume for each sample, and it will just concentrate and then move the EVs after concentration to your output plate or to the columns. This instrument works with the plates, but also it works with the columns. So if you're doing plates, you can run up to 2 plates, so 48 samples. If you're running columns, you can run up to 24 columns. So 24 samples, so half of the throughput. It's a fully automated instrument. You're just putting the stuff, pressing start, and you're done. And it does the concentration as well. And here on the right, there are actually 2 different wash bottles, because as we said before, typically people clean the columns with NaOH followed by PBS. So here on the Summit, it's doing it fully automated for you. So you just tell it, I want to finish the run with washing the plates or the columns with NaOH and buffer, and the Summit will wash it for you, and the columns or plates will be ready for the next experiment. And this is how the deck looks. So we have here the input plate. So again, it can be either a plate or tubes. So just Eppendorf tubes. You have here the position for the 2 plates or the column holder. So up to 2 plates or up to 24 columns. Then you have here 3 positions for the 3 fractions, which again all sit on top of chambers for vacuum, so you can really concentrate your samples on it. And then the instrument will move the samples to the output plate or to tubes. So it can be either a plate or tube holder. And for the concentration, so there are papers who compare different filter for ultrafiltration concentration. People typically use the Amicon filters. And so we have RC 10K filters. This is what typically people use in the field. Happy to share with you a paper where they compare different cutoff, different membranes, and they found out this is the optimal one. But we have other versions as well, PES filters, so we can just accommodate your needs. And just a little bit about the summit performance. So again, we really think about reproducibility because this instrument is for running clinical samples. So you can see how tight the CVs are within a plate and the plate-to-plate CVs. Moving from the Ascent to Summit has like similar performance. So again, you can optimize your assay with the low scale on the columns. Then when you're ready for the 1,000 samples, you can move to the Summit. And the concentration can give you up to 10x concentration by concentration. You can get higher fold increase in volume, but there is always loss to the filter. But you can get around 8 to 10x concentration of your EVs, including the loss. So that was the Summit instrument for fully automating your sample isolation, EV isolation. And just going back to our vision, which is to really look at the entire workflow of isolation, how to do it automatically, and the analytics. So I showed you our Atlas EV ELISA, which what we call it a QC ELISA. It's more of like, instead of running a Western blot, which takes overnight and has less sensitivity and does not work for total plasma, you can just run this Atlas ELISA. For people who want higher sensitivity, we released last year a more sensitive and wide dynamic range version of this assay. We call it the Atlas Lumi ELISA, because it's based on chemiluminescence readout. So you can see here the comparison between the two. The orange is the absorbance one, which is around 10x less sensitive and has only 2 log dynamic range, whereas the green one, which is the chemiluminescence, you can measure samples with a lot of EVs and low amount of EVs on the same run together with the same dilution factor. We also have a homebrew kit for the chemiluminescence assay. So let's say you want to capture a specific target on EV, we can give you a conjugation kit, so you can look at a specific EV transmembrane protein. And we're now working on I always use the wrong button. We are working on the next generation analytics pipeline. If you have interest in collaboration on that, please contact me. So we are developing a very sensitive method to colocalize markers on the EVs using proximity assays. And the idea is to really look at 2-3 marker combination which are associated with a specific tissue or disease. I'm interested in neuro diseases, but we're looking at cancer and other diseases as well. That's it. Thank you all for coming with all the different circumstances. And I'm happy to take more questions, and also feel free to reach out you have my email about anything I presented. Okay, let's see what we have here. Do you have those 24 sec plates with multimodals? And that's a great question. We do not currently, but we will have them by next year, we hope. So currently we only have the plates with the 4B and the 6B. And this is why we made sure the Summit can also run columns in case you want to run the MM as well. But we will have the MM plate version. It just requires more optimization to make sure it's like fully reproducible. And so we're taking the time there. Okay.
I think it's pretty much it, yes.
Awesome.
Yeah, it's look amazing, and I want to say everybody that we have on June the conference of EVTech,
yeah.
and Tal is representing there, and Tal have exhibition over there, so you can feel free to ask question and to come to see the exhibition as well.
Yeah, no, I'm very excited to come in June. Hopefully to meet many of you in person. I will give a talk and I will have a booth and I will bring the Ascent with me as well. So yeah, really looking forward to seeing you all in person in June.
Just a few more questions, Tal. They just came on the chat. Any loss of EVs during concentration by UF?
Yeah, so you always have some EVs sticking to the filter. From my experience, actually, the more the EVs were pure to begin with, the higher the losses. So if you use the MM column, then the EVs are very pure, so you have slightly higher loss. I've seen up to 30% loss of EVs in that case. And this is why I'm saying that although I can concentrate by volume around 15-fold, the actually effective concentration is then 8 to 10x, taking into account this loss. That makes sense, Moshe? I'll take it as a yes. I don't see you.
Sure, he agrees.
Yes. Okay, awesome. And yeah, I said before, the multi-mode columns are reusable. I explained before the trade-off and a little bit about the lipoprotein carryover, but we tested them 3 times. And I think there's a question for Dimitri about washing the stained EVs. I guess this is for FACS or for
that would be another step of colon if you want to do it, or you do it before. You can do it on the EVs before the isolation to get rid of it.
Yeah, actually, so you have 2 options. You can either, after you stain, use the Vesicles columns or other versions to clean the EVs. But we also wrote a white paper with particle metrics showing how you can stain the EVs before the SEC. And then the SEC will both separate your EVs from contamination and from the free antibodies. So, but this requires to stain the actual sample, which the downside is you need to use much more labels because you're staining the full sample. But you can look in our website on our work with particle metrics, and we show there how you can stain before the SEC.
And handling the EVs before FACS. I think it really depends on I don't think there's anything particular. They need to be probably concentrated after the ascent or the summit, just to get a lower volume, which is always better for staining. That's what I would recommend anyway.
Yeah, typically people also they concentrate, stain, and then they dilute them out. So they're diluting the labeled antibodies, or you can use a cleaning method. Awesome.
Any more
thank
questions?
you all for coming. I think we're good, right?
I think it is.
Awesome. Thank you for having us, Avi. It was great. Thanks for all the great questions.
And see you in June.
Yep.
Thank you very much. Bye-bye.
Bye.
Good day.